In the last lecture, we talked about Mendel's monohybrid cross and his first law of genetics, which is that individuals have two alleles of each gene, but when they form their sex cells, those genes separate. So a sex cell only gets one allele from each parent. There are some other types of common genetic crosses besides the monohybrid cross. So the monohybrid is a cross. We'll actually do it in lab, but other types of crosses are also used in genetics. And so some other types of crosses. One, and we'll see this one in the future too, is the test cross. The test cross is a cross between a phenotypically dominant individual and a recessive. A test cross is where you have a phenotypically dominant individual, but you don't necessarily know the genotype. The genotype of a dominant is unknown. So I'm just going to use a generic symbol here. So we've got a dominant individual. We'll give it a capital letter and uppercase letter, indicating that it has one dominant allele. But we don't know if it's homozygous or heterozygous. So a test cross is a cross between this dominant and a recessive. What do we always know about the genotype of a recessive? We know the genotype of a recessive. It is homozygous recessive. So the dominant and recessive. If we look at the outcomes of a test cross, what if the dominant individual was homozygous? What would be the result? Well, you could throw yourself together a quick Punnett square. Now, oftentimes students will, separate the alleles and do this. And this will work, but you don't have to do 4 squares. You can just do 2. What do we end up with here? They're all dominant phenotype, and they are all heterozygous. But since these gametes are the same, we don't have to, because if we put the frequencies in here, this would be 1/2 and 1/2, and this would be 1/2 and 1/2. But if you take these and add them together, this is 1, right? So you could also do this Punnett square just like this, because all the gametes are going to get a big A, and all the gametes from the other plant are going to get a little a, so it's all that. So the results of a test cross with a homozygous dominant is that you get all dominant individuals, offspring, and they're all heterozygous. But what if the dominant individual is heterozygous? So what are the results of a test cross with a heterozygous individual? Well, so let's look at a Punnett square. Separate these alleles. So that's what Mendel's first law says. The recessive only gives you 100% of the gametes produced by the recessive. 100% of the gametes are going to be little a. 1/2 for the heterozygote will be dominant, and 1/2 will be recessive. So what are our results? 1/2 will be big A, little a, and the other half will be little a, little a. The results of a test cross with a heterozygote are a 1-to-1 ratio, phenotypic ratio, where half are dominant and half are recessive, and a 1-to-1 genotypic ratio. So the test cross is a really common tool, not necessarily in humans, mammals, in organisms that you don't cross very well, but in organisms that you can manipulate like plants, fruit flies, nematodes, and different genetic model organisms, test cross are quite common. Another type of cross that is used is called a back cross. So the back cross is the cross of a hybrid with a parent. So it's a cross of a hybrid, so an offspring with a mixed genotype with one of the parents. Another type is the reciprocal cross. In a reciprocal cross, well, these are used to determine whether the sex of a parent influence influence the inheritance pattern. And we'll look at circumstances where this does happen. So in a reciprocal cross, we use two different crosses where the sex is reversed in the second cross. What do I mean by that? This is best done by example. So if we have a male who is dominant for a trait, so let's say YY, and the female is the homozygous, this is one cross, right? The reciprocal of this would be a male who was recessive for the trait and a female who is dominant for the trait. So this is a reciprocal cross. And sometimes where sex does influence the inheritance pattern, or if we have sex-linked genes, the sex of the parent does matter in the outcomes. Now we're going to take it up a notch. So we looked at how one type of gene, one trait or one characteristic passes on to an offspring. But Mendel didn't stop with a monohybrid cross or with a one-factor cross. He continued on and he looked at how do different traits, how do the inheritance patterns of different traits occur? Do they influence each other or are they distinct from each other? And in doing this, Mendel derived his second law. So Mendel's second law is the law of independent assortment. It's the independent assortment of genes. Different genes, I should say. Independent assortment of different genes. And I'm going to put an asterisk here or a star, and I'm going to say this law only applies for unlinked genes, because we'll look at linked genes in the future and may violate Mendel's laws. So this is only for genes that are not linked together. So this is Mendel's second law, independent assortment of different genes. And so to do this, he did two-factor crosses. Where he studied the inheritance pattern of two distinct characters or traits or distinct genes. So let's look at an example. So our example, what do I mean by different characters or traits? So in an example here, we're looking at shape. Oh, we've got a round seed and we've got a wrinkled seed. So I'm using the same characteristic or the trait that we looked at before. So we're going to use seed shape. Our distinct variation is again wrinkled versus round. So that's one characteristic or trait. Then the second one that we're looking at here is seed color. So now we're looking at a distinct characteristic or trait, and it is seed color with variations of green and yellow. All right, so using the same concept of a P generation, he started with a pure breeding green round seed and a pure breeding yellow wrinkled seed. Now this is just an example. He could have, he did this in all sorts of different forms, but the P generation is still the P generation, meaning that it is pure breeding. So pure breeding plants, which we know genotypically now means homozygous plants. So we've got a round green and we've got a wrinkled yellow. So the premise of this is to see, does the shape of the seed influence the color or vice versa? Are these independent traits? Are these traits completely independent of each other? So the results that he got from this were that in the F1 generation, they were all round and yellow. What does that mean? Where did the green go? It seems to be masked. That would suggest to me then that green is a recessive trait, right? And so if this is a pure breeding and homozygous for the green trait, and if this is recessive, let's give it, what symbol do we want to, we'll call it little g. But we know that that green is recessive and that yellow is dominant because the yellow is masking the green. And all the seeds are round, so that means that the round characteristic is dominant. So we'll use that same symbol we used before. And that the wrinkled is recessive. So if we look at these genes, we've got the R gene and we've got the G gene for the shape gene and the color gene. Notice that this individual is homozygous for the round and homozygous for the green. This is still pure or true breeding because all of the offspring of a self-cross would result in round and green seeds. And this would all result, a self-cross would always result in wrinkled yellow seeds. So what we're seeing here, so in determining dominant and recessive, we're still having the dominant allele masks the recessive allele. But this doesn't tell us whether or not green influences yellow or wrinkled influences round. So we didn't stop here. We went on and did, and we're going to call this, by the way, this is a dihybrid. The dihybrid, meaning a mix. So the hybrid means a mix and di means of two. So it's a mix of two traits. So round and wrinkled together and green and yellow together. It's a dihybrid, a mix of two traits. So what happens when you take one of these round yellow individuals and cross it with another one and collect data for, dozens of offspring? The F2 generation resulted in, and we'll look at the basis for this. So I'm just going to kind of skip ahead. So it resulted in a distinct phenotypic ratio of 9 to 3 to 3 to 1. So the results of a dihybrid cross results in a 9 to 3 to 3 to 1 distinct phenotypic ratio, where the 9 is the category where they're dominant for both traits. So in this case, we'll say big R dash, because we don't know it's in the second position, and big G dash. 3 out of the 16 total would be dominant for one characteristic and recessive for the other. Another 3 out of 16 would be vice versa. And this one lonely 1 out of 16 would be recessive for both characteristics. Here's a figure on the next slide to kind of show you the same cross. And I think we use the same symbols here. So they're in a little bit of a different order, but here's what you can see kind of the outcomes look like. And we're going to go ahead and we're going to put together this Punnett square. And so I'm going to start you with it. We're going to look at the formation of the gametes, and then I want you to cross-check your work with what you see here. So the conclusion from this 9 to 3 to 3 to 1 ratio, and we'll see why that is in a second. The conclusion of this 9 to 3 to 3 to 1 ratio of a dihybrid cross was Mendel concluded or devised the principle or the law of independent assortment. Where different genes do not travel together to the gamete. They assort independently. Okay, so let's take a look at what this means. So if we take our round green seed parent had a genotype of big R, little g, little g. This was our round green, right? When we cross it with the wrinkled yellow, which had recessive for one, dominant for the other. So Mendel's first law says that these have to separate, right? So big R is going to separate, but the gametes are going to get one of those R's, and the little G's are going to separate. So all the gametes are going to get a little G. So in this case, for the parental generation, 100% of the gametes are going to get a big R and a little G, because these can't travel together. These are alleles of the same gene. They can't travel together, they separate. That's the first law. The second law we're going to illustrate here in a second. But for these ones, these also separate. The gamete produced by the wrinkled yellow, they're all going to get a little R, and they're all going to get a big G. So when they come together, when the pollen and the ovum comes together, the result is a big R and a little R, and a big G and a little G. This is the dihybrid. This was the F1 generation in Mendel's cross. But now we have to look at this and see, where does the independent assortment come in? So do the big dominants like to hang out together and go to the gamete together, or is it a random process? Is this one just as likely to go with the big G as with the little g? Or do the dominants hang out? Or does it like the recessive better? This is what we're going to look at. And so if these independently assorted, how would the gametes form? And that's what it really comes down to. If these independently assort, how would the gametes form? We know big R and little r have to separate. So we know that one half of the gametes are going to get a big R. The other half of the gametes are going to get a little r. Now, if the assortment was independent or random, if big R was just as likely to go with big G or with little g, then the outcomes for the gametes would mean half of the time it would go with big G, or half of the time it would go with little g. And the same for the little r. Half of the time it would end up with big G, and half the time it would end up with little g. So our gametes, the gametes that result, if these were independently assorting, we could take our frequency, multiply them together. 1/4 of the gametes would be big R, big G. Another 1/4 of the gametes would be big R, little g. 1/4 would be little r, big G. And the other 1/4 would be little r, little g. So that would be the gametes that would be produced by the dihybrid. These would be the frequency in which they were produced. Now, in an F1 and a dihybrid cross, we would have some of this stuff here. Erase this so we can make a little Punnett square to the right of it. So if we took and did a dihybrid cross, these would be the gametes produced by each of these individuals, right? Because they have the same genotype. So we can set this up in a Punnett square. And actually, let me go back and erase this again and set up my Punnett square right here. So these would be the gametes produced by one parent. And then the gametes produced by the other would be in the same frequency because they have the same genotype. So 1/4 would be this. Another 1/4 would be big R, little g. Another 1/4 would be little r, big G. And 1/4 would be little r, little g. Now, when you combine these, if we look at all the potential outcomes, by the way, let's look at how many squares are here. 4 by 4, this is 16 possible outcomes, 16 possible ways these gametes can come together. And so when we write the genotypes of the possible offspring, we write the alleles of the same gene together. So this would be big R, big R, big G, big G. This would be big R, big R, big G, little g. Big R, little r, big G, big G. Big R, little r, big G, little g. So these are the possible genotypes in the first row. What do we have here? We have round, yellow, we have round, yellow, we have round, yellow, we have round, yellow. They all have that dominant phenotype. So like I said, go ahead and finish off that table yourself and then count up how many are round and yellow, because that's the dominant phenotype. And what you'll see is there's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them are dominant for both traits. So again, in this case, RRGG. 3 are green and round. Another 3 are wrinkled and yellow. And 1 out of those 16 squares is recessive for both of the characteristics. So is the double recessive, little r, little g, little g. Now this happened like that because big R was just as likely to go with big G as it was with little g. The genes are not attached. They are not physically together. It's a random sort of process of what goes to the gametes. And we're going to talk about the basis for this in terms of chromosomes. I just want you to look at a chromosome for a second. Here's a karyotype of a human. So this is not what you would see in a pea plant, but just as a depiction, if you took all the chromosomes from a human, chromosomes from a somatic cell, they exist in pairs, one from mom, one from dad. Well, the genes are physically located on the chromosomes. So if we say gene A, for example, has a physical location, also called a locus, here on chromosome 11, you have 2 copies of those. That's Mendel's gene pairs. And if you're heterozygous, you would have 2 different versions of those genes. So if you were heterozygous, they're both gene A, right? They both code for the gene A trait, but if they're different, they have different phenotypic outcomes, potentially. The different characteristics, the different traits. So the round versus the yellow, again, this is a human genotype or karyotype. Different genes have different physical locations. So for example, this is where the human ABO blood type locus is located. We inherit 1 copy from mom, 1 copy from dad. This is where one of the globin, one of the genes that codes for hemoglobin is located here. 1 copy from mom, 1 copy from dad. There are different genes, but you might have different alleles of the same gene.